Hello, and welcome to today's webinar, organised by the Pathologist and Ultiview. My name is Luke Turner, Associate Editor of the Pathologist, and I'm delighted to be the moderator of today's online event, which will explore how to improve the accuracy of PD-L1 diagnostic tests with multiplexing and image analysis. Please allow me to introduce today's speakers, Dr. Angela Vasaturo and Dr. Matt Humphries. Dr. Vasaturo is a Senior Field Application Scientist at Ultiview. In 2012, she was among the first postdocs in Europe to be involved in the development of multiplex immunohistochemistry and multispectral imaging and analysis of up to 6 immunofluorescence markers. Today, she is considered a leading European expert in multiplex IHC techniques, multispectral digital pathology, and tissue imaging. Dr. Humphries is the scientific lead for tissue hybridization and digital pathology in the Precision Medicine Centre of Excellence at Queen's University, Belfast. His research interests are focused on the digital pathological assessment of immune biomarkers in solid tumours and the application of AI in pathology across both research and clinical diagnostic settings. Today, Dr. Humphries will discuss whether the application of digital image analysis and multiplex immunofluorescence to digital PD-L1 immunohistochemistry slides has the potential to improve the accuracy and reproducibility of PD-L1 diagnostic tests. Both of our speakers will be on hand to answer any questions that you may have at the end of the presentation, but please remember that you can submit questions at any time throughout the webinar. So, without further ado, let me get today's webinar underway by passing you on to our first speaker. Dr. Vasaturo, over to you.
Thank you, Luke, for the introduction, and good afternoon, everyone. As we have heard, today I'm going to be joined by my colleague Matt Humphries from Queen's University, Belfast, to discuss how we can improve the diagnostic accuracy of the PD-L1 test with fluorescent multiplex and image analysis. Before discussing in depth multiplex IHC, I would like to go through the evolution of histopathology and the different types of immunostains that are used to analyze morphological features, cellular structures, and different cell types. The most popular of the staining method for routine diagnosis is what you see on the left panel, hematoxylin-deusin staining. H&E reveals structural information and is broadly used to assess cellular and morphological features, identify the type of tissue, and also, of course, pathological changes. The use of the H&E has been the most effective and most common procedure for pathological diagnosis of cancer because it allows pathologists to pinpoint focal areas of a tissue specimen containing aggressive tissue and foster a proper diagnosis. Next to the H&E, we can now add in a single protein detection with the chromogenic IHC standard method, which you see in the second panel in the middle. And of course, depending on the type of marker we use, we will be able to identify a cell population. However, with regular IHC, it is actually impossible to assess co-localizing markers, especially at the single cell level. And this is because when you look at multiple markers with this type of technology, the stainings are performed on consecutive sections. And this, of course, radically limits an accurate classification of cells that require detection of multiple markers. This limitation is overcome by multiplex IHC, which now allows to look at multiple proteins rather than only one, and therefore allows a deeper cell type characterization and a better understanding of pathological processes like cancer. Then, on top of this, being fluorescence, we can also add the range of expression of each of the biomarkers. And since we are looking at multiple cell subtypes, we can also assess their special interaction. So how do we do that? This is an overview of the technology that Ultiview has developed. So let's have a look at the chemistry itself. We use primary antibodies that have been conjugated with the DNA barcodes, and each antibody has its unique DNA barcode. This allows us to perform just a single antigen retrieval step, and of course, this helps keeping sample integrity. So after the one single retrieval step, we perform a single staining step where we apply all the antibodies at once. And this, of course, reduces the staining time because you do not have sequential staining of each of the primary antibodies you use, and eliminates the need for secondary antibodies. It also gives unlimited capacity to colocalize markers on the same subcellular compartment. So the next step after the primary incubation, the staining step, is the amplification step. This is basically a linear amplification that happens at the same time and same rate for all the antibodies that we use in the incubation primary step. This allows also the identification and quantification of a broad dynamic range of expression, and it happens by elongating the DNA sequence of each of the antibody. And this increases the number of binding sites for what we use in the next step, which is the detection step. So we use a mixture of fluorescent probes, the detection step is actually based on a DNA hybridization technology. So the fluorescent probes that we use are made of a DNA sequence, which is complementary to the barcode sequence, which is on the antibody, and the fluorophore. We use only 4 classical fluorophores that are spectrally distinct, which are FITC, TRITC, Cy5, and Cy7. And this is because we want to avoid spectral mixing. Now, there are multiple advantages of using the InSituPlex technology, and I'm going to show you just a couple of them. So the first one is that we can perform whole slide imaging. And this allows us to extract all the data from all the cells in each sample. This is particularly important when we work with tumor samples that are already highly heterogeneous. And of course, once we have the whole slide image, we can still annotate region of interest, and then we can decide if we want to perform analysis on the entire tissue sample or just on the annotated regions made by pathologists. One of the major challenges of multiplex IHC so far has been to enable multiplexing on different biomarkers while retaining the advantages of the H&E in terms of morphological information. So the Ultiview InSituPlex technology allows now the combination of H&E and fluorescence staining on the same exact slide. So here you see an example of non-small cell lung cancer that has been stained in the upper image here with the fluorescence, so with 4 markers, 5 colors. And then in the lower panel, you see the corresponding H&E performed on the exact same slide. So if we zoom in on a higher magnification region of interest, you would see here the fluorescence staining, which includes CD8, CD68, PD-L1, and pancytokeratin, which is the kit that also you will hear about in the next presentation. And when you look at the exact same region stained with the H&E, you can appreciate that there is no morphological changes, no difference between the tissue morphology in the 2 different images. This means that the type of assay that we perform is really gentle and does not change your sample morphology and allows the downstream analysis after your multiplex IHC, which is something really unique. So how do we do that? We have established an automated workflow that you see here in this slide, which gives same-day staining to results and includes 3 main steps. The first one, of course, is the staining, which now takes only 5 hours and a half. And with the Ultiview technology, you do not need any type of assay development or optimization of your panel. They are all ready to use. So once you have performed your staining, then you can move to whole slide imaging. And depending on the tissue size, this can take around 15-20 minutes. So you can do this whole slide imaging with any type of scanner fluorescent scanner that you already have available, as long as it can read the 4 classical channels, FITC, TRITC, Cy5, and Cy7. And then the same day, you can now move to image analysis with any image analysis software that you already have available in your lab. So the initial UltiView offering has been built around the T cells due to their central role in the immune response to tumor, which is, of course, mediated by the interaction of those T cells with other immune cells like macrophages, myeloid-derived suppressor cells, or dendritic cells, etc. We currently have available different ready-to-use multiplex kits that answer the main biological questions around cells and pathways that are crucial in responses to tumor and therefore are also targets of immunotherapy. These include what you see in the first row, the T-ACT kit and the APC kit to look at the T-cell activation and that professional antigen-presenting cells that guide their activation. Then in the second row, you see the MDSC and the Treg kits that help to look at the immune suppression in the tumor microenvironment. And in the last row, the PD-1 and PD-L1 kits that are made to study T-cell exhaustion and tumor evasion. Now, being PD-L1 the focus of this webinar, I'm going to show you a little bit in more details what the PD-L1 in UltiMapper IO kit includes. So this includes CD8, CD68, PD-L1, and the tumor marker, being in this case pancytokeratin, and the nuclear counter stain, of course. With this panel, you can basically identify all the cells that express PD-L1, either immune cells or tumor cells, and then the cytotoxic T cells that express CD8. Here I'm showing you an example staining of the use of the PD-L1 kit, which has been done on non-small cell lung cancer. So here you see in green CD8 cells, and in the second image you see PD-L1 expression on tumor cells that express cytokeratin, where cytokeratin is in cyan and PD-L1 is in red. And then next, you see PD-L1 expression on macrophages. And in this image, you see CD68 in yellow and PD-L1 in red. And here you have a general expression of PD-L1 on both T cells, macrophages and tumor cells. And here emerged image showing all the different markers. So I have shown you now the kits that we have available on the market, but we have recently moved, we have transitioned from a fixed content that you have seen in those kits to a more flexible content. So now you can pick your markers of choice and you can build your own custom kit containing up to 8 markers. So you are now able to stain your samples with more biomarkers, up to 8. And even in this case, or even if you increase the number of biomarkers you are staining on your tissue, you are still able to keep the same sample integrity. And I'm going to show you here an example on a sample that has been stained with a 12-plex panel that we have developed and we currently offer in our services lab, which includes the main markers of immune cells. And here again, it's a non-small cell lung cancer whole slide that you see on the left side. You see the 12-plex multiplex IHC image. In the middle, you see the same slide, H&E. And then in the last image, you see the fusion of H&E and fluorescent image. If we zoom in on this sample and we look at one specific region of interest, here you have all the markers and you can easily define what is immune cells and what is tumor cells. Then if you look at the corresponding H&E, which is what pathologists are used to look at, they would be really easily able to say what is tumor and what is in tumor infiltrate. But what if you can add on top of this the information about what type of cell infiltrates you have in there? Like, for example, in this case, you see that the majority of cells are stained with this light blue, which is CD20. So it's mainly B cells rather than T cells, for example. So I would say that currently, histopathological tissue analysis by a pathologist really represents the only method for confirmation of presence or absence of disease, for the identification of hot and cold tumors, for example, or disease grading or measurement of disease progression. Therefore, allowing pathologists to add precisely on an H&E slide the subtype of infiltrating cells stained by multiplex fluorescent IHC can deliver more accurate prognostics and patient stratification for treatments. So integrating the cell-level information that you have with the multiplex IHC with the context-specific information of the microenvironment that comes from the H&E. And with this, I'd like to thank you all for your attention, and I'll pass it on to Matt Humphries to hear about how he has applied the Ultimapper IO PD-L1 kit to his research.
Hello, everybody. Thank you for that introduction. I'd just like to thank Angela as well for that fantastic overview of the UltiView technology. It's going to save me quite a lot of time. So I'm going to talk to you today about a study that we've done that was published in Cancers recently. And this was on improving the diagnostic accuracy of the PD-L1 test with multiplexing and image analysis. Now, I don't need to tell a cancer research audience the importance of the PD-L1 checkpoint in cancer immunotherapy. We all know that the binding of PD-1 and PD-L1 inhibits T-cell activation that allows the immune suppression and can potentially cause neoplastic growth. What many people might not be aware of is the myriad of companion diagnostic assays that are available for the PD-L1 diagnostic test. These come from multiple vendors. They are multiple clones, usually tied to automated staining platforms and, unsurprisingly, to the compounds and reagents and drugs that these companies sell. Some of them actually have different scoring methods. In fact, PD-L1 SP142 has just been accepted as a clone of choice for the diagnostic test in triple-negative breast cancer, among others. And this scoring method is slightly different to the rest, which do talk a similar language in terms of tumor cell-only expression. So we published a paper back in 2018 on this and identified a new role for PD-L1 predicting good outcome in HER2-positive ER-negative breast cancer. I'd encourage you to check that out as well. But the one thing that these diagnostic assays do have in common is the need for the subjective pathologist assessment. So to give you a little bit of context about the situation in Northern Ireland, within the Precision Medicine Centre, we house the Molecular Diagnostic Service, which delivers molecular diagnostics for the whole of Northern Ireland. And I'm going to talk about our experience with the Ventana Roche SP263 clone, as well as the multiplexing that we've done within this study. And our experience with reflex testing is just over 3 years now, and we've conducted reflex testing in well over 3,000 non-small cell lung cancer cases. We actually originally described this cohort and the reflex testing that we do in the Current Standards and Future Opportunities paper we published in the Journal of Thoracic Oncology, and this paper actually lays the groundwork for quite a lot of this study. So again, I'd encourage you to take a look at that. So if you're unfamiliar with the assessment of PD-L1, these are the gold standard rules and procedures that a pathologist should follow when they're assessing PD-L1. So there needs to be a minimum of 100 tumour cells within the sample. We would ideally like no strong background staining. And what you're looking for is partial or complete linear membranous staining on the tumour cells alone. So only viable tumour cells are considered positive. So, T-cell expression, you've got to discount any macrophages. So the scorer needs to consider the number of positive tumour cells in the sample over the total number of cells in the sample. And the decision to treat or not treat is at 1%. So anything less than 1% is negative, 1 to 49 is a low positive, and then you'd be a candidate for second-line therapy. Anything over 50% is a high positive. And the clone SP263 is predictive of response to Pembro or Keytruda, if you like brand names. So the materials that we would receive are resections, biopsies, and FFP cell blocks. And I've put that little caveat in there that membranous macrophage staining is extremely common in non-small cell lung cancer, and even to the most trained pathologists can be very, very troublesome to discount sometimes. So some of the challenges that we routinely see in the diagnostic service are low cell number. So sometimes there's a need for an additional stain, such as a TTF1 looking at adenos or a CK56 for squames. And this is really just to convince ourselves that there are at least 100 tumour cells within the sample. Again, beneath that on the bottom left, you'll see a feature that we've termed the hugging effect, and this is where the PD-L1 positive signal in the stroma is actually hugging a this tumour epithelial nest is perhaps best seen in this negative case here. So you really have to be unequivocally sure that the cell is either positive or negative, the tumour cell, before you actually include it in the score. And this can be very, very difficult when you've got sporadic single cell staining within the tumour. As I've said, macrophages can be extremely difficult to discount sometimes. So if we take a look at the top centre, you'll see a PD-L1 positive cluster. And when you look at the H&E, you could be forgiven for thinking that is epithelial cells. But actually, when we look at the cytokeratin, it's negative, and the CD68 confirms it as positive macrophages. So that cluster does have to be discounted. Weak staining is also an issue. You'll see in the top right-hand corner at the times 40 magnification, there is an actual positive cluster of cells there, but unfortunately the staining is so weak, the pathologists sometimes have to rack up and down between the focal planes to actually detect those very fine membranes. So this is a real problem for image analysis when you're looking at a 2D image as well. As well, the heterogeneity of PD-L1 is an issue. When you're looking at a large resection which contains hundreds of thousands of cells and tens of thousands of tumour cells, these small foci of PD-L1 positive cells across the section need to be counted up, and it can be extremely difficult to determine whether or not all these cells together make up at least 1% of the specimen. This is something that digital pathology can help with massively. So the goal was to apply digital pathology to our routine IHC and the multiplex slides to try and bring reproducibility and accuracy to the test. And there were several solutions that we employed to try and achieve this. So the first solution was quite obviously to apply digital pathology to the routine IHC slides that we had. So we took all the manual scores that were determined clinically by trained pathologists, and we compared this to a digital pathology score determined in QuPath. Now, the vast majority of these scores were concordant, and you can see those with the green dots. There were some cases that were discordant, which were in red. And really, most of these discrepancies were around the clinical thresholds of 1 and 50%. And this was mainly due to poor staining, which the image analysis couldn't cope with, or these overabundant intrinsic macrophages, which were sometimes overwhelming the classifier. And then sometimes this low detection threshold in the very fine membrane staining cells could also sometimes be missed by digital pathology. But you'll notice that there are some blue dots within those. And this was where we were actually unsure what the ground truth was. And this is because the subjective nature of the assessment of PD-L1 is extremely difficult. So we could have a pathologist that comes along and says the ground truth of this case is 53%. So that is in the high category. The patient would get the first-line therapy. Now, image analysis, or you or me or another pathologist could come along and say, well, actually, I think this is 97%. It's still correct. It's in the same clinical category, and the patient would have no adverse effects. However, if the program or you or I or another pathologist said, well, actually, I think this is 49%, it's actually closer in percentile points, but it's in a different clinical category. And so technically it's incorrect according to the first assessor. Now, we actually don't know which is the right answer here. So this is where the ground truth becomes a little bit fuzzy. So in some instances, we actually didn't know which was true and which wasn't when we were comparing manual and digital assessments. And here are just some examples of QuPath actually undertaking the image analysis of PD-L1. And this really is just to say that it's easy when it's easy. So if you've got very clean resections, or in this case, biopsy specimens, the image analysis program is quick to identify the tumour, the stroma, and the fact that there are no PD-L1 positive cells within the red area of the tumour. And again, the strongly positive cases are the same, so it's easy when it's easy. Sometimes when you do have this heavy macrophage infiltration, as you'll see in the second image, we have to train that additional classifier to try and tease out those different cells, because those macrophages are PD-L1 positive and could confound the assessment. But where digital pathology really does have a huge benefit is in this sporadic single cell staining across large resection specimens. So instead of having large areas of clear PD-L1 staining, these cells are often dotted throughout the specimen. And as I say, if you're looking at tens of thousands of these cells to actually quantify these in and around a threshold is extremely difficult. So again, this is where digital pathology is extremely useful. So the next solution is multiplexing. And here we have a PD-L1 reflex tested case. And this is a non-small cell lung cancer strip biopsy. And if we take a look at the times 40 magnification in the bottom right-hand corner, what you can see is an epithelial cluster of cells. And within this, what looks to be a positive foci of PD-L1 cells. So this case was scored clinically at around 20% across the whole specimen. But then what we did was we took the UltiView Multiplex PD-L1 IO kit, which has been excellently described previously. Again, this was fantastically easy to use, went straight on the Bond RX, and then when the slides came off, we imaged these on the Vectra Polaris. And then, so taking a look at that same clinically reflex-tested case, you can see here in the multiplex image a fantastic delineation of all these different cell types. So what we're looking at in green is the cytokeratin, but the PD-L1 in red, CD68 is the macrophages, and then the DAPI is staining all the nuclei. And you can take a look at the composite image in the top right-hand corner of the screen, and immediately what you can see is that the red PD-L1 signal is actually coming from these intraepithelial PD-L1 positive macrophages. So when we actually did the quantification by eye again, we thought, okay, there's been quite an overestimation there of PD-L1 positive score, but we still think it's in the same clinical category, and we scored this much, much lower. So this is now a tool that we can bring to bear on PD-L1 clinical testing. It's a tool we can offer to the pathologists, where we can quickly stain these images and present them to the pathologist on screen, and the pathologist can turn on the individual channels, pan around, zoom in, and look at areas of interest and be unequivocally sure that the score that they're reporting is as good as it can be. Now, a lot of people say this is all very well and good, and it's at this point they say, well, fair enough, but you still need a pathologist to sit at the screen and assess these cases, and that's absolutely true, you do. So one of the things that we wanted to do was try bring image analysis in the same way we did to the IHC slides to the multiplex slides. And there are several solutions that we've explored to do this. One of the most advanced solutions that we have at the minute is a machine learning approach actually built on the Polaris, which we have used a train by example approach where we've shown the program hundreds and probably thousands of cells at this point and just positive, negative, and it's learned over time to assess PD-L1. And in the case that you've just seen before, it predicted that the accuracy of positivity of PD-L1 was at 2% in this case. We're also working with Visiopharm as part of a UKRI project on a one-button press workflow where we can just press go, or the pathologist can just press go, and we will have annotations, cell detections, identification of the tumour, all the phenotypes that are possibly available within the sample. And then we can apply really advanced AI to remove things like tissue folds or necrosis, anything that could confound the assessment. And then in this proof of principle on that same section, again, we were getting an accuracy of around 2% for the score. So very encouraging. But if you know anything about Belfast, you'll know that we routinely use QuPath in our digital pathology workflows. And if you're not familiar with QuPath, I couldn't recommend it highly enough. It's absolutely fantastic. The brainchild of Pete Bankhead, and it's an absolutely wonderful tool, and we use this every day. So the vast majority of our image analysis is being through the QuPath workflow. And when we're discussing IHC, again, if you're familiar with QuPath, you'll be familiar with all these steps. But just in this example, I'm discussing tissue microarrays here, but you would bring in your image, you would annotate it or de-array it if it's a tissue microarray, and then you would annotate the tissue itself and go through quite an advanced quality control step. Now, this is probably the most crucial step in image analysis, is making sure that you remove all the things that could potentially confound the final score, whether that be a TMA score or an overall case, so things like tissue folds, abnormal structures, necrosis, whatever it might be. Then you can do your cell detection, which detects all the viable cells within the sample that you want to assess. In this particular instance, we would then do a tumor stroma classifier, and then within the tumour itself, we would look for the PD-L1 positivity, and then we can export that data and compare it to the manual assessment. Now, when it comes to multiplexing, the steps are very, very similar in that you obviously import the image, you de-array or annotate if you're not dealing with TMAs. Again, the intense quality control is absolutely essential. You can detect the cells, and this would be done with the DAPI channel in this instance rather than the hematoxylin. The principles are the same. And then you can either use the default parameters within QuPath to detect the different channels and the different cell types, or as we tend to do is use bespoke scripting to pull out the different phenotypes and different combinations that could be in there and export that data, and again then do the comparisons. So very, very quick to be able to batch process dozens and dozens of slides at a time. But one thing that we were, I suppose, hesitant of going into multiplexing is the reliability between the IF and the DAB IHC. So what we did was we took a few tissue microarrays that we had available to us using non-small cell lung cancer TMAs, and we took sequential sections of these and stained with DAB IHC and the Multiplex kit, the Ultimune PD-L1 IO kit. And what we've seen is, and just taking a few examples again around these clinical thresholds, is a fantastic correlation between the singleplex DAB IHC, and when we take a look at the single channels, the exact same expression is also seen. So I'm not really discussing phenotypes here, and is this cell expressed within the tumour or in the stroma, or are the combinations of PD-L1 positive CD68 macrophages here or there. That's not what the purpose of this was. This was assessing the comparability of DAB IHC to single channel multiplex IF. And here are some of the images from that assessment. So in column A, we're just really looking at PD-L1. So the top two panels are looking at the DAB IHC. So the first row at the top is the native IHC image, followed by the QuPath image analysis mask. And so we've done a tumor stroma classifier for PD-L1, and then within that, the black cells are the positive PD-L1 cells. CD68, we didn't do a classifier, so anything black in that is just positive CD68 cells. Likewise for CD68, and again, we use cytokeratin for our tumor stroma, so on the far right, you're just seeing tumor stroma classification. In the third row, this is the native single-channel IF. And again, for PD-L1, CD68, CD8, and cytokeratin, the image analysis masks were very, very comparable. So when we compared the data extracted from the DAB IHC images and the single IF channels, we found a fantastic correlation between all the different channels. So we were highly confident in the comparability between the different methodologies, and you can see the R-squared values there. So we were very, very confident taking this forward that the multiplex immunofluorescence would give us a comparable assessment to DAB IHC. And here we have a native image of all the channels turned on for the multiplex for this particular kit. And again, the QuPath mask, which is the mask that's the bespoke scripting applied. So this now allows us to interrogate the tumour microenvironment very, very specifically. So when we take a look at the clinical question, which is we're only really interested in PD-L1 within cytokeratin positive tumour cells, if we take a look at B1 and the exploded view, what we can clearly see is that there are PD-L1 positive cells in the tumour epithelial cells. So these are the numbers that we want to extract and compare to the clinical assessments or the digital pathology assessments. But as I've said, that we can have these confounding cell types within the sample, and B2 identifies the CD68 macrophages which can plague the assessment. So the white arrow is actually pointing to an intrinsic PD-L1 positive macrophage inside the nest. And again, this would be a data point that would be excluded from the analysis. And then we also have macrophages that are outside the tumour nest in the stroma, which may have prognostication and is something that we're investigating in our research questions. But for a clinical assessment, we definitely need to discount the intrinsic macrophages inside the tumour nests. As well, CD68 can be PD-L1 positive, and again, in the bottom right, we can see on the white arrow inside the epithelial nest, there's a CD68 positive, PD-L1 positive cell. We need to make sure that's completely discounted from the analysis because obviously that's not a PD-L1 positive cytokeratin positive cell. So if you think back to the digital image analysis that we did, I mentioned that there were several discordances that we identified. And what we did on sequential sections was to apply multiplexing to those cases. And what we found is that we were actually able to rescue the discordant cases back to what was clinically assessed. And this was only possible with multiplexing. And really, we believe the reason for this was because multiplex was able to better delineate the different cell types within the sample over and above what could be done by image analysis. And as I said on the previous slide, from a clinical perspective, we're only actually interested in PD-L1 and cytokeratin expression in the tumor compartment, but the different phenotypes that are available within the tumor and the stroma could have implications for prognostication that we're actually exploring in some of our research questions. And so what we proposed at the end of the Kansas paper was a case triage workflow whereby we would say that 100% of the IHC slides would be submitted to image analysis de novo, and when you're not near the clinical thresholds of 1% and 50%, we were actually, with a high degree of confidence, able to determine the PD-L1 score accurately, in line with the pathologist's score. Now, when we got close to the clinical thresholds, we did need a manual review of the digital image analysis, and if there was still some diagnostic uncertainty, what we did was we multiplexed those, and then multiplexing, again, was able to recapitulate the pathologist score extremely well. So our proposal is from 100% of these slides going into this particular workflow, the pathologist would really only need to manually review the digital image analysis in about 5% and about 1% of the multiplexes. So to conclude, I hope that I've shown you that the diagnostic assessment of PD-L1 is extremely difficult, but digital image analysis can be a reliable method of case triage, especially when you're not around these clinical thresholds, which alone can save a massive amount of pathologist review time. Hopefully I've shown you that digital workflows can be fast and efficient, and that multiplex facilitates this rapid digital diagnostic interpretation, and it's highly accurate. It can be a simple process to be able to quantify truly positive epithelial cells within the sample, and really does diminish the need for this subjective manual scoring. And if you've read the paper as well, you'll definitely see that we've fallen down heavily on the side of UltiView and the Ultimap IO kit as the multiplex of choice in comparison to the DAB. And this wasn't just for reasons of concordance as well, but when you're applying this in a clinical laboratory, you need to consider things such as the complexity of the test, which was very, very easy, the component reagents to prepare, again, this is a simple cocktail as you've seen, the fewer retrieval steps required, and the speed of the automated staining run. So you can do this within one day, very similar to a diagnostic IHC run, whereas some multiplex protocols require 12 to 14 hours plus. But crucially, there was no need for this complex in-house pre-validation. So again, other multiplex methodologies, you need to consider the antibody concentration, the antigen retrieval, secondary balancing of the fluorophores, and all this can take weeks and weeks and months in some instances to work up these panels. So it was a no-brainer for us when we're thinking about this diagnostically. So to conclude, multiplex IHC, the next generation of molecular pathology? I think it potentially is. But we'll have to wait and see what comes from clinical trials that are currently underway that are assessing multiplex IHC in these critical questions. But I think multiplex is definitely on the horizon for molecular pathology and molecular diagnostics. So with that, I'd like to thank everybody within the Precision Medicine Centre of Excellence, particularly the Director, Professor Manuel Salter-Tellez, and the Co-Director, Dr. Jackie James, who's also the Scientific Director of the Northern Ireland Biobank, who were instrumental in providing these tissue samples to us. Far too many people to mention, so I'll just leave it there. I'd also like to thank the funders and you for your attention. I'd be happy to take any questions. Thank you.
Thank you, Dr. Vasaturo and Dr. Humphries, for that interesting exploration of digital image analysis and multiplex immunofluorescence in the context of PD-L1 diagnostic testing. We've received a number of questions from audience members, so we'll try and get through as many as we can in the time remaining. Please be assured that any questions that are not answered live today will be answered via email. Okay, first question. How do you deal with tissue autofluorescence?
I'm going to take this question at least partially.
Okay.
Yeah, yeah, and then I'll pass it to Matt to hear his personal experience. So there are a couple of things I will mention in terms of reducing tissue autofluorescence. The first one is that most of the background signal that we see when we stain FFPE slides comes from the non-specific binding of secondary antibodies. And with the Multiplex technology, we do not have this issue because we do not use at all secondary antibodies. And the second thing is that we actually use dyes that are quite bright, and therefore we have a very high signal-to-noise ratio. And therefore, also when we scan slides, the the exposure times we use are really very low, so you don't see at all, almost at all, the autofluorescence. And Matt, you can add your experience on this.
Yeah, I'd agree with Angela on that. I mentioned in the talk that we have a Vectra Polaris, and while that is capable of spectrally unmixing if you're using different multiplex technologies, that's not something we really needed to do with Opal because of everything that Angela said. So we actually found the autofluorescence less of an issue when we're using the UltraView kits.
Thanks both. Next question asks, how feasible is the deployment of this technology in wider clinical practice, especially thinking about small regional hospitals?
Yeah, I'll take a run at this, Angela, if that's okay. This is a question that I always get asked when I give this talk, and it's a valid question because the deployment of this technology is very, very specialized, and for me, I see this being something that's delivered within centers of excellence such as ourselves. Not only do you need the specific technology in-house, and the kit itself is not cheap, let's say, and then you obviously need the specialist pathologist training on hand as well. So for small regional hospitals, I don't see this being something that's easily deployable, at least in the current situation. So I would say that centres of excellence are definitely the way to go for multiplexing, especially with regard to clinical practice.
Great, thank you. The next question asks, how long does the scanning step take? And could you also remind the question asker what scanning you use, please?
Yeah, I think I just touched on that in the question before. We do have a Vectra Polaris in the PMC. And scanning is quite variable, actually. It really does depend on what the size of the tissue that you're scanning, and as well the number of channels that you would assess. So it can take anything up, from 10 minutes to 30 minutes to do a good scan. And again, obviously, you would need to QC that, and sometimes the need to repeat it. But I would say no more than 30 minutes.
Thank you. The next question is in regard to the analysis of multiplexed IHC via fluorophore labeling. How much of an issue is the interference of fluorescence channels with overlapping excitation frequencies?
With regard,
I'm going to take this and
yes,
oh, sorry.
Yeah. No, by all means, go on.
Yeah, I just want to mention, then I'll pass it to you for your personal experience. But I mentioned also during the presentation that we have really chosen for dyes that do not have any overlap. So we do not need spectral mixing, and we do not have any issues with the bleed through of signal. So we use the 4 channels that are really spectrally distinct. So FITC, TRITC, Psi5, and Psi7.
Yeah, and for us, this is something that we do in our QC steps on the scanner itself before we then bring it into image analysis. But it's, as Angela said, there's great spectral separation there, so we can accomplish just by simple thresholding the ability to just pick out these different channels quite easily.
Great, thanks. The next question asks, what are the fluorescent channels used when moving to Hyplex, so more than 4 markers?
I guess for me again. So once more on the fluorescent channels we use, we still keep using the same ones. So when we move to higher level of plexes, or more markers, let's say 8 markers, the staining, let's say it takes exactly the same, more or less the same time. So, and it stays the same way in the sense that we still have only one retrieval step and we incubate all the antibodies at the same time in one single step. And what differs is actually just the detection phase. So up to the amplification, all stays the same, with also the amplification that happens at the same time, same rate for the 8 markers. And then the detection happens in 2 times, because again, we want to keep using only these 4 fluorescent channels that do not have any overlap. So we will take a first image with the 4 channels and then dehybridize the fluorescent probes and go in again in a second run with the fluorescent probes that are complementary to the second set of primary antibodies and take a second image. So we will have 2 rounds of imaging with the same exact fluorescent channels that then we will need to overlay, so to co-register.
I don't think I have anything to add to that. I fully agree with what Angela said.
Great, thank you. Next question, what is the process for ABS optimization and validation?
For me again. We basically always start by selecting when there is a new antibody entering in our UView biomarker menu, we always start screening 3 different clones. And our gold standard is still the DAB. So we will perform DAB staining with the regular antibodies, and then we will conjugate the antibody with the DNA barcode and repeat the DAB staining. So then we can assess for specificity and sensitivity of the antibody and if there would be any effect of the conjugation on the performance of the antibody. And once this is done, then we move to fluorescence with a singleplex and then a multiplex building up a panel.
Great, thank you. Can you comment on any differences between using QuPath and Visiopharm for PD-L1 analysis?
Yes, I can. So we tend to use QuPath really because we're extremely familiar with it, and the main differences is in how specific and detailed you want to get into the image analysis. So I love Visiopharm; it's a fantastic tool. I would say that you're probably better going down the app route and going direct to Visiopharm for that, because to actually build the app yourself, you have to be very, very knowledgeable about the different pixels that can be manipulated in the image, and then you have to detect the certain lines and the masks that would then be applied. But what Visiopharm does have above QuPath is that integrated AI tool that I touched upon. So I would say that the main differences are that QuPath is much easier to use for an entry-level image analyst if you've just got an image and you want to do simple cell counts. But if you're looking at very nuanced and very small specific features in and around cells, then Visiopharm might be something that you want to explore.
Thanks, Matt. Next question. You mentioned having the HE on the tissue. Is that an artificial HE or is this an actual HE stain on the same tissue?
No, it is an actual H&E. So we first perform the fluorescence staining and then we perform a regular H&E as you would do on any other slide. So it's not an artificial H&E.
Great, thanks. Can this procedure be done manually? And if yes, what are the challenges?
Yes, it can definitely be done manually, and not big challenges in the sense that it takes exactly the same time, 5.5 hours, including the retrieval step and even the DWAX. yeah, pretty straightforward, just few steps in the protocol and no big challenges. So it can be done manually, easily.
Thanks. Someone has followed up to a previous answer that you gave. It's about the fluorescence channels. Just to follow up on the question, does this put a limit on your experience on how many multiplex markers can be mixed together at all?
Well, routinely, and now what we offer commercially is up to 8 markers, even though, as I shown during the presentation, we offer in our services lab a 12-plex. And actually, we have done a proof of concept going up to 16 markers, which means 4 rounds of detection step. The point is that basically we want to use those kits in clinical trials, for example, and we want to use it on, we aim to use it routinely, as Matt has also shown. So we really believe that we do not need so many markers to really use those this multiplex IHC routinely. So in principle, we can go very high, but we like to be placed in what we call the validation space. So we are not really a discovery platform where you want to use 40, 50 markers at the time. We really want to have a deep understanding at the single cell level and validate the relevant targets that can possibly be applied into the clinic.
I would agree to that. I'll just add slightly, just to say that you know, assessing a couple of biomarkers in a single image just visually is exactly what I think is needed to be able to tease out all the cell types in the sample. And if you start going to that much higher number, then you do need to bring image analysis in, and that definitely becomes more of a research question rather than a clinical question.
Great. Sorry for cutting a question there, Matt. I was just going to say we're getting lots of great questions coming through, so keep that going. Thank you, everyone. The next one asks, can you strip and restain? So, for example, if we have stained with your PD-L1 kit, can we redo the same slide with the 12 multiplex?
So once more, we do not do any type of stripping, and when we go higher than 4-plex, we basically just use multiple rounds of detection with the same fluorescent channels. That said, basically when you want to go higher than the classical 4-plex, now we have the UView biomarker menu where you can pick and choose your markers, and then we build up the kit for you. But again, at the moment, it's just up to 8 markers. And the 12-plex panel is just offered by our services lab.
Thanks. Do you annotate and cut out necrotic areas to rule out an unspecific staining with PD-L1?
Yes, absolutely. It's essential to do that. In fact, I tried to labour the point quite hard in one of the slides there that, you know, this is a critical stage in the analysis that you have to do extremely rigorous QC. I did allude to the fact that there are some AI tools out there that can assist in this, but it's absolutely essential to take out those necrotic areas because you can get that non-specific stain in it. So yes.
The next question asks, are there any AI tools used during diagnosis?
Not currently within the Precision Medicine Centre at Queen's, no. We are contributing to the Pathology consortium, which aims to develop AI tools for potential diagnostic application, but currently there's no AI used during the diagnostic practice at Queen's, no. So it's the Belfast Social Health and Care Trust within the Precision Medicine Centre at Queen's.
Great, thanks, Matt. I'm afraid we're going to have to leave it there. Again, let me reassure you that any questions left unanswered today will be answered after the event by email. This webinar will also be available on demand in just a few days' time, and you will all receive an email alert when it is ready. Thank you once again to Dr. Vastachuro and Dr. Humphries, and thanks to everyone for joining us today. I look forward to seeing you at future webinars. Goodbye.